Hey guys, this is Mrs. Allgood. Today we are going to do a little biochemistry review. And I know some of you might be thinking, if we only have 2 free response questions, why are we reviewing the content? Well, it may ask you to apply some content that you know. I don't think it's going to be so easy that you could do it without having sat in the class all year. I don't know though. And some of you may be taking like the biology SAT subject test in June, if that gets up and running by then. Either way, it's not going to hurt you to review. And to me, this is time better spent than trying to learn new content that we know is not going to be on the exam. We know biochem's fair game. And even if it's not asked on your 2 free response questions, it's going to help you in the long run to be able to review this. So, kind of up to you if you just want to listen to this or you want to take some things down as you go. This just happens to be a flip chart that I already had made. So I'm just going to be talking through it. You're fine if you want to write some things down or if you want to just listen. Okay, that's fine too. All right, first thing we should start with is a functional group review. This should be something that you remember. You've got to know what these functional groups are. So let me switch over to my typing. Remember that a methyl group is going to be CH3, and that 3 is going to be a little subgroup. Hydroxyl is an OH group. Sometimes you might see this with the dash or the bond in front of it because the oxygen is going to be what attaches to the rest of the molecule, not the hydrogen. A carbonyl group is a C double bonded to an oxygen. And remember, 2 subtypes of carbonyl groups are aldehydes and ketones, and those are just different based on where that carbonyl group is in the overall molecule. If it's at the very end of a molecule, that carbon might be attached to a hydrogen, and then we're going to call that an aldehyde. If it's more in the middle of the molecule, we're going to call it a ketone. Sulfhydryl is just your SH group. Phosphate, PO4. So, here's something to think about with phosphate groups. Like we did the phosphorus cycle. When you see phosphate groups, you should automatically be thinking of things like the phosphate group in DNA or phospholipids. Like you should be able to figure out, okay, what kind of molecules that we've talked about this year might have this group or might have these elements. All right, this one I might just hand draw. This is a carboxyl group. So this is going to combine a carbonyl group with a hydroxyl group. So it's going to be a C double bonded to an oxygen. And that same carbon is also going to be bonded to a hydroxyl group. Okay, and then an amino group is usually just NH2. If it's ionized, it might be NH3+. Okay, so those are your functional groups. Next, let's go through our 4 major categories of organic macromolecules. So you should be able to list those. Okay, that should be something like there's a lot of things in bio that should just be like word association. If we say 4 macromolecules, you should think carbs, proteins, lipids, nucleic acids. Okay, don't get caught up in so much detail that you aren't able to tell these apart from each other. Okay, so on this slide, we're just going to go over some major differences between them. Okay, carbohydrates contain carbon, hydrogen, and oxygen. Okay, Lipids, for the most part, contain those three elements also. So the difference is going to be in the ratio and also how they look. Okay. Carbohydrates are always going to have twice as many hydrogens as they do carbons and oxygens. And that means that the number of carbons and oxygens in carbohydrates is going to be fairly equal. And we'll look at specific categories of carbohydrates, but many of them come in a ring type form. We'll look at some specific categories on the next slide. All right, lipids. Lipids also mainly contain carbon, hydrogen, and oxygen, but they contain much less oxygen. Still usually twice as much hydrogen as it does carbon. That's pretty standard. But usually lipids are going to have a lot less oxygen than your carbohydrates do. All right, proteins. Remember that proteins are probably the most important of our four macromolecules as far as, serving the needs of biological organisms. So remember that proteins are made up of amino acids, and they will contain the elements carbon, hydrogen, oxygen, and nitrogen. And then nucleic acids. Nucleic acids are made up of nucleotides. So nucleotides are going to be the building blocks of nucleic acids. And just keep in mind that nucleic acids are things like DNA and RNA. And as far as the elements that nucleic acids contain, they're going to contain carbon, hydrogen, oxygen, nitrogen, and then remember they're going to have phosphorus because of the phosphate backbones in the sugar phosphate backbone, because of those phosphate groups. So I'm not saying that you have to copy down this whole thing. I think that's a little much. I don't really feel the need to post it either because you already have notes on this. This is just refreshing your memory. So jot down what you feel like you need to, but you don't need something else to print off at home right now. You just need to listen and to hear just a refresher on some of these important topics. Okay, so we're going to look at the three categories of carbohydrates a little bit more closely here. All right, so our smallest category of carbohydrates is going to be our monosaccharides. Okay, monosaccharides, I'm going to move this thing down there. Monosaccharides, you've got several examples here. You've got glucose, you've got fructose, and you've got galactose. Now, keep in mind that monosaccharides tend to be able to take on one of two appearances. They can be a simple ring sugar, like you see here, or they can be a straight chain monosaccharide. Okay. What determines which form they take on basically has to do with how humid or how much moisture is in the environment at the time. Besides glucose, fructose, and galactose, keep in mind that a couple other important monosaccharides are ribose and deoxyribose. Okay. Your next category is going to be your disaccharides. Disaccharides, we've got some of those listed here. Remember, those are going to be two rings joined together. So some disaccharides will include sucrose, lactose, maltose. Those are the three that I normally have you be aware of. We did learn about oligosaccharides. Oligosaccharides usually have, between about 4 and 6 ring sugars there. And then once you go beyond oligosaccharides, then you get to our larger sugars, which are our polysaccharides. Okay, polysaccharides, we can split these into plant polysaccharides and animal polysaccharides. Okay, plant polysaccharides are going to be starch, which remember, starch is for storage. I prefer typing to handwriting because this does not work that well on my computer. Okay. Another plant polysaccharide is going to be cellulose. Okay, we find cellulose in plant cell walls, and that is going to be mainly for structure. There we go. So, starch is for storage, cellulose is for structure. Now, as far as our animal polysaccharides go, we've got glycogen. Okay, glycogen is going to be for storage. Glycogen, we store away excess sugars in our liver and muscles as glycogen. So once you've used up the glucose in your bloodstream, this is going to be the next place that we can tap in order to access some sugars. All right, and then structure, we have chitin that makes up the cell wall in fungi, and it also makes up the exoskeleton of arthropods. Okay, and then as far as what polysaccharides look like, you can take a look at this. Look at this picture here. See how it's made up of many rings joined together? That's pretty typical of a polysaccharide. Okay? All right. Okay. Now, let's talk about dehydration synthesis versus hydrolysis. Okay. So dehydration synthesis, occasionally you'll hear that called a condensation reaction, and that's because in condensation, water is normally formed. And that's what happens here. So remember, we can do dehydration synthesis with any of our macromolecules and their building blocks. So let's say we were doing this with carbohydrates. If we were to, we could do dehydration synthesis between two monosaccharides in order to join them together to make a disaccharide. Okay? So in order to do dehydration synthesis, this is going to involve taking a hydrogen off of one of our monomers and an OH off of another monomer, and you link those together to form water, and then water is going to leave. Okay? And then that links the two monomers together. And so notice here I have something about entropy. Okay? And what I'm asking there is what's happening to the entropy when we go from this to that. We're taking two separate things and we're combining them to make one larger thing. So what we are going to be doing to the entropy here is we are decreasing it. Okay? Because remember, entropy is the amount of disorder. So the more pieces we have, the more particles we have, the more disorder that we have. Here we are reducing that disorder because we're combining two smaller pieces together to make one larger piece. Okay, let's look at the opposite of that. That's going to be hydrolysis. So remember that the term hydrolysis literally means to break with water. So here we are adding water in order to break a larger molecule apart into two smaller ones. Okay? We are going from one larger thing to two smaller things. So as far as what this does to entropy, well, we are increasing disorder. We are increasing the number of particles we have. So we are going to say that this increases the entropy here. All right, lipids. Sorry, I forgot how I had this laid out. Lipids, we have three major categories. So remember that lipids are going to mainly have carbon, hydrogen, and oxygen. And they probably will have twice as many hydrogens as they do carbons. but the oxygen is going to be much less. So notice you just have oxygen a little bit here. So your first category here is going to be your fats or triglycerides. Okay, and so you've got two categories of those. You've got your saturated fats and your unsaturated fats. Okay, so what you see in the upper left here, this is a saturated fat. It has saturated with hydrogens, which means that given the number of carbons in each of these fatty acid chains, it has the most hydrogens that you could possibly stick on there, and that's because all of the carbons are just single bonded to all the other carbons. There's no carbon-to-carbon double bonds. So for saturated fats here, remember what fats and triglycerides are made of. They are made up of a glycerol, which is this part over here, and then starting with that carboxyl group, this is a fatty acid chain, this is a fatty acid chain, and this is a fatty acid chain. So saturated fats, we say, have no carbon-to-carbon double bonds, which means there are no kinks in their chain. So because there are no kinks in their chain, they tend to be fairly straight, which means they can pack tightly together, which means these are going to be solids at room temp. Unsaturated fats, remember, a fat is also called a triglyceride. And so the tri part is due to the three fatty acid chains. So in order for something to be considered an unsaturated fat, it needs to have a carbon-to-carbon double bond in at least one of the three chains, which this one here does. Okay? So these do have carbon-to-carbon double bonds, which means they have kinks, which means they are, chains are spaced further apart, which means these are going to be liquids at room temp. All right. The second category of lipids here, let me draw in my little zigzags. Okay, your second category here is going to be your phospholipids. And your phospholipids have two major portions, okay? They have a head region, which is polar, and the two tails, okay, that are non-polar. So we say that this head region is hydrophilic because that end likes water because it is polar. Polar things mix well with other polar things. So that means that, well, I'll show you the cell membrane in just a second. But that means when we're looking at phospholipids, that means these heads, okay, should be facing a water-based fluid like the cytoplasm or the extracellular fluid. So we say that this is hydrophilic polar phosphate head. Okay, that means the two tails are going to be hydrophobic, nonpolar fatty acid tails. So they're fatty acid chains just like these, okay? Why do we care? Hold on, let me go back. Sorry, let me go back. Even though it's not letting me. Let's look for phospholipid bilayer. All right, this one will work. All right, sorry about that, guys. I'm back. So what I wanted to show you with this, because I know I've given you some FRQs in the past about this, or maybe it was multiple choice, I don't remember, is remember that here's your layer of phospholipid heads, phosphate heads, and then there you have the layer down there as well. What you got to keep in mind is that whatever fluid is up here, that when these phospholipids orient themselves, the heads automatically are going to orient themselves towards a water-based fluid. And that water-based fluid could be cytoplasm, it could be the extracellular fluid. Okay, but those are both water-based. So the phospholipid heads are going to want to be facing there. Okay, your nonpolar hydrophobic fatty acid tails are going to want to be facing away from those polar fluids. Okay, so this is going to be a very hydrophobic interior to the membrane. All right, so that's phospholipids. Let's keep going. Then we have steroids. Okay, so steroids are 4 linked carbon rings. So 1, 2, 3, 4. Remember that anywhere you see a point here, it is understood that there is a carbon. If other atoms are not shown, it is understood, like, so for example, let's reduce the size there. So like there is a carbon here, and it is understood that that carbon is single bonded to this carbon and is single bonded to that carbon. And then we have a bond to this carbon, and then there's this. So it's understood that yes, there is a C at the end of that line, but it's understood that that is a CH3 there. Okay, not that that's super important for the AP exam, just keep that in mind. One thing that we'll review probably sometime next week is remember that you have 2 different kinds of hormones. You have steroid-based hormones and you have protein-based hormones. Keep in mind, we talked about this a couple times this year, that the receptors for the steroid-based hormones would be located in the cytoplasm, because that means that that steroid hormone can pass through the lipid bilayer and bind to receptors that are in the cytoplasm. So some examples of steroid-based hormones would be things like testosterone, estrogen. Okay? Proteins. Okay, so keep in mind, proteins, I didn't want to do that. Proteins are I'm trying to write on, there we go. There's my little text box. So proteins are made up of amino acids, and that those are linked by peptide bonds. Okay. Levels of protein conformation. Let's find a picture, shall we? That will do. Okay. So 4 levels of protein conformation. You've got your primary structure, which is just your sequence of amino acids. Okay, so description, Again, I don't expect you to be taking notes on this. You already have notes on this somewhere. But we would just say that this is a sequence of amino acids. Important bonds here. Well, remember what kind of bonds we said link amino acids together? Those are peptide bonds. Oh, one other thing we were supposed to do on this one is I want you to think about the structure of an amino acid, the generic structure. You've got a central carbon in the middle. You've got a hydrogen up here. You've got a carboxyl group here. You've got an amino group here. All right, and then hanging down from that central carbon is going to be your R group. Okay, now R does not stand for any certain element or anything. R is the rest of the molecule. It's what makes one amino acid different from the others. Some of those R groups can be polar, some can be non-polar, and that's why when you think about mutations, okay, when we learned about different categories of mutations, when you change an amino acid in the sequence, okay, you are changing possibly its chemical properties. You could be going from a polar amino acid to a non-polar amino acid or vice versa, and that will affect how the protein folds at that tertiary level, which means it's probably not going to fold into the same shape that it once was, which means it's probably not going to behave the way it once did. So going back to this, let's go back to our picture. Secondary structure can be an alpha helix or a beta pleated sheet. And so we would say that new hydrogen bonds form, causing the polypeptide to become an alpha helix or beta pleated sheet. Okay? There we go. See if we can get that all to fit there. So important bonds here would be your hydrogen bonds. Okay, when we get to tertiary, okay, notice that a tertiary, remember I used to show this to you like a phone cord, and that phone cord could be folded on top of itself, and at some regions you might have an alpha helix, and other regions you might have a beta pleated sheet. But a tertiary structure is going to be it's going to be one polypeptide chain folded on itself into a specific three-dimensional shape. Okay, and we talked pretty extensively about some of the different bonds that hold these together. So of course, you're going to have your hydrophobic interactions. That's where all the non-polar amino acids like to get together. Remember, I used to say it was like me at a party, being with all the other antisocial people. You're going to have some ionic bonds. You're going to have some hydrogen bonds. You're going to have disulfide linkages or bridges, and that's between a cysteine usually and another cysteine, since cysteine is the amino acid that has sulfur in its side groups. Now, as far as quaternary structure, remember this is when I would get multiple phone cords out. So this would be two or more polypeptide chains folded together into a specific shape. And really no different bonds here than any of the other levels here. So what that would look like is if you look at something like this might be hemoglobin, where you have 1, 2, 3, 4 different polypeptides. So technically it's anywhere between 2 and 4 polypeptide chains folded together into a specific shape. Okay? So that is it for our lecture today. Like I said, if this seems kind of awkward for you, it's a little awkward for me. I don't really mind teaching in this format, but reviewing is just real different for me. Because I'm trying to figure out the review method that works best, and I know how to review when the exam is in a traditional format. But with the exam format being changed, you know, I want to review the content with you, and I want to do it in the best way possible to help you. So when people say, well, what's the best way? What do I need to review to be absolutely prepared for the AP exam? I can definitely give you suggestions. It's really not going to hurt you to go do, you know, some of the practice stuff in your Strive for a 5 workbook that you got in the beginning of the school year. I'm going to try and give you a lot of FRQs as time goes on. Remember, we're still over a month away from the AP exam. Okay, so the FRQs are going to get more intense as the time goes on. Right now, we're just trying to ease ourselves into a way of reviewing. So don't worry, I don't really want us to jump right into the hardest FRQs when we're still a month away. I want us to work our way up to it. I want to look at some terminology with you and figure out a review method that works best. All right, until next time, guys.